it's after 4, so let's go ahead and get started. For those of you who don't know me, my name is Gina. I'm a professor in the department. Thanks for coming to the EEB seminar series. It's my absolute pleasure to introduce you to Ruth Hufbauer, who I've known for a long time, and I completely forget how we met. Twitter? Okay, a good Twitter friendship. So Ruth is a fantastic evolutionary ecologist who uses applied systems to ask basic evolutionary questions or foundational evolutionary questions. She asks questions about population expansion and what allows a population to adapt versus go extinct. I think today she is part of our diversity seminar speaker, or she's the diversity seminar speaker. So she's going to give us a seminar in 2 parts, one in which she's going to tell us a little bit about her work as the Fort Collins or Colorado State lead of their NSF Advanced Grant, and then some of her science. So let's welcome Ruth.
Thanks. Is this working? Can you hear me? Too much? Too little? Good? Okay. Thank you so much, Gina. It's really a pleasure and an honor to be here. I've had so much fun today chatting with all sorts of people. And like Gina said, she invited me in part for my role working on DEI at the university that I'm at, Colorado State University. So I'm going to talk a bit about science and then a bit about that. And I had so many good scientific conversations today. I'm like, oh, I wish I were talking about this project, oh, that project. But basically, I'm just talking about the one that I have already prepared. I resisted changing the slides last minute. So I'm going to do that first. Is this working still? This is no longer working. Maybe it's my phone. Oh, which I was going to set a timer on. So, say again? Oh, it did. It went. All right. Okay. So I'm going to start off talking about the role of density dependence in evolutionary rescue. And I want to acknowledge these fabulous collaborators who I've worked with on this, as well as NSF funding. And since I'm not talking about other work that I've done, I just want to say, if any of you ever want to talk about tarantula ecology, right now I have somebody doing her PhD on tarantulas in my lab. It's so much fun. I'm learning a ton. Arthropod food web ecology on tropical islands, biological control of weeds. I've worked a lot on biocontrol of weeds and using them to understand invasions and range expansion dynamics and the evolution of dispersal on range expansion fronts. I have a student who just finished on some population genomics of butterfly adaptation to high elevations. Anyway, there are lots of those fun systems that are out in the wild, and I'm going to be talking about an experimental evolution system with a model system in the lab that I also really love. So there we go. In addition to acknowledging those collaborators, I want to particularly point out Laura, who was on that first page. She was a postdoc in the lab overseeing the collection of most of the data I'll be telling you about. And she worked with a bunch of fabulous undergrads for this work. So when environments change, and they are changing very rapidly in the modern world, populations that have previously been relatively well adapted to the location they're in can find themselves strongly maladapted. And this, there's a lot of theory around what happens there. So if the environmental change itself is super severe, population growth rate can drop below the replacement rate. And if you don't have any evolution happening, those populations will just go deterministically to extinction. But Gommel, Kaiwitz, and Holt proposed that if adaptive evolution is rapid enough, the populations could grow again. And so this is what they called evolutionary rescue. So I'll use this term evolutionary rescue, but it's really just rapid adaptation in the context of environmental change. It's not anything other than that. And I also just want to really quickly acknowledge Dick Gomel-Kaiwitz. I don't know if anybody know Dick in this room? He's a wonderful human being who unfortunately died all too early. But he was great as an experimentalist like me to work with his theoretical stuff. I could just call him up and talk ideas with him, and he would run different simulations for me and think about different things. He was just so fun to work with. Okay, so a big question in this area, maybe I'll step up here so I don't trip over that, of evolutionary rescue is, like, what are the contexts that will allow populations to adapt rapidly enough so that they don't go extinct? And then understanding more about that, then are there things that we can manipulate about populations to help them persist through environmental change? And so people have studied evolutionary rescue a lot in the lab, like I'll be showing you. And so it's been shown to happen in yeast, in bacteria, in Microsoft error reporting. I just want you to go away. And so, like, yeah, this can happen. This is great. But these populations are starting, like in this really elegant experiment by Bell and Gonzalez, they started with over 500,000 individuals. So these are massive populations. And oftentimes when we're worried about populations going extinct, we're worried about animals and plants that have much smaller population sizes in the field. And so some of the work that I've done in my lab is using this beetle as a model system to kind of bring one level of kind of closer to systems that we might be concerned about to some of these questions. And so here we showed that you can get this characteristic U-shaped curve of evolutionary rescue even with an organism that is diploid and obligately sexual, which means that there's going to be inbreeding happening and inbreeding depression. It also, obligate sexuality means that demographic stochasticity is going to be a whole lot greater. So even in a system like that, you can see super rapid evolution that helps populations avoid extinction. Hello. I feel like I need to be closer. So these experiments and others like them have shown that evolutionary rescue can happen. But one thing that has been true of most of these is that they have maybe not avoided, but the experimental design has been such that strong negative density dependence isn't part of the situation. So we know things about evolutionary rescue, like genetic diversity enhances rescue, which makes sense, more diversity, more evolution. That has been explored pretty well in these, but we don't really know about negative density dependence and how that could influence evolutionary rescue. And this is really important because density dependence is super common. And I just want to, before I get into that, well, no, this is a different slide. Sorry, this is exactly this. So density dependence happens with small animals that have large populations. So this shows the growth rate of mosquitoes given different larval densities, showing that growth rate decreases with density. So that's negative density dependence. So it's very commonly shown experimentally with small animals and large populations. Large animals with much smaller populations, you can see similar patterns. It's really common in plants, and that's true even when the plants have very small populations themselves. So you don't need to read this, but it just says neg, 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 neg. So that's whenever there is a little asterisk, that means there's significant negative density dependence due to intra-specific competition in these really small, rare plant populations. So negative density dependence is super common, so it's important to understand how it might influence evolutionary rescue. So that's the main question for the experiment I'll show you. And this is really in working in this space of kind of eco-evolutionary dynamics that I'm sure you've probably all heard about. So it's asking, like, how do ecological factors, in this case we're talking about something really basic, density, population density, influence evolution, and then how does evolution influence ecology? And for the moment, we're really talking about adaptive evolution, but you know, it doesn't necessarily have to be adaptation. So that's what I'm going to examine in this system. So I mentioned Tribolium already. It's a little tiny beetle that feeds on flowers, so not flowers that you give your sweetheart, but flower that you, sometimes you have to unplug it and replug it. I will try that. That you make your cookies with. Aha. And so here's its habitat in the wild. Grain silos, essentially. And here is its habitat in the lab. We can grow it in these little boxes where we have media that is like flour mixed with yeast. And we use non-overlapping generations in our experiments just because that makes it more tractable for combining with modeling. And their generation time is short, but nothing like a yeast or a bacteria. It's 5 weeks. 5 weeks can feel very long if you're doing multiple generations. So what we did to look at the role of density dependence on evolutionary rescue is a factorial experiment where we manipulated density, diversity, and evolution. And I'll go through each of these in turn, but basically they each had 2 levels, these factors. And our challenging environment was just adding an insecticide to our media with flour and yeast. Oops, wrong way. So for density, all we did was maintain the same habitat size. So if a population doubled in size, the habitat size stayed the same, and so density doubled. Whereas for density independence, if a population doubled in size, the size of the habitat doubled. You can imagine that as populations grow, that like quickly becomes really challenging to manage. The other thing I want to point out, I have independence in quotes here because, so we had 10 beetles per little patch, and so density was low. Tribolium can experience a Lee effect, so positive density dependence. There shouldn't be much positive density dependence at that density. There's going to be weak negative density dependence. So it's not true density independence, but it's as close as we can get experimentally. But it's this low constant density that even as populations grow, density doesn't increase. And so we used populations for these density treatments that were either diverse populations or had passed through a pretty strong bottleneck in population size. And then we had them either evolving or not. And that's preventing evolution is hard when you have something that's obligately sexual, but you can do it. And I will show you how we did that. So for evolving populations, we've just got your basic descent with modification. Parents give rise to offspring that then become parents, etc. So continuous line of descent. For non-evolving populations, we have those parents that give rise to offspring that we then stick in the freezer and replace them one for one. So like if there were 2 offspring, we replace them with 2 individuals from an ancestral population that is not exposed to the pesticides. So selection, they're not experiencing continuous selection by this pesticide. All right, and I'll go straight to the results in a moment. So this is what this experiment, just the expansion of density independence was kind of nightmarish, but Lore is amazing, and yeah, we managed it. So first thing is that density dependence strongly increases extinction risk, and that is both for diverse populations and bottleneck populations, with the difference being larger for diverse populations. So makes sense, right? Makes biological sense. Populations are size is being constrained by negative density dependence. They can't grow as much, and so more of them are going extinct. And then for population dynamics, it's pretty fascinating. So first, let me show you this key. We have diverse populations are in the bluer colors. Bottlenecked populations are in yellow and red. Higher density, so density dependence, is the darker color for both of those. So a key thing is that bottlenecks reduce that population growth. They're really limiting population growth. So the density, even the density independent populations that are bottlenecked, they have as much space as they need. They're not able to really take great advantage of that space. And then density dependence also really strongly limits population growth from the density independent to the density dependent in both the diverse and bottlenecked groups by different amounts, but still strong limitation for both of those. And that also makes perfectly good biological sense, right? As if with more space, with more habitat and less negative density dependence and competition, cannibalism that's going on, the populations are able to grow in size. The thing that's amazing is how evolution plays into this. So if we just move that key over to the side, we can look at the non-evolving populations. And so you can see that this wild population growth is only able to happen because the populations are adapting and evolving. So I think when we see invasive populations in a new environment and their population sizes, they become really enormous. We always have to be keeping in mind that it's not just that this environment's great for them. There's a ton of rapid adaptation that's going on that if they couldn't do it, they would be staying down here like this. And I think something really fascinating is that the density, having the combination of density dependence and a bottleneck gives population dynamics that are essentially like no evolution whatsoever. That doesn't mean evolution isn't happening, but you can't see it in the population dynamics. Okay, so at the end of this experiment, we evaluated fitness in a common garden environment. So we looked at realized fitness across a range of densities from the low density that we used in the density independent treatment up to 100 is much lower than the high density, how high the densities got, but at least there's a lot of negative density dependence going on there. And so this uses those same colors for evolving populations here with the solid lines and non-evolving populations. And you can see that like a ton of adaptation has happened that's allowing these populations to thrive in this insecticide habitat. Unsurprisingly, diverse populations are better able to adapt than the populations that went in through a bottleneck. And there's this hint of potential adaptation to higher density with the negative density dependent populations having slightly higher realized fitness, but there's a ton of uncertainty in those estimates. So the other thing I want to just point out here is that just because we can't see that adaptation in the population dynamics curves, it's happening. They're just not able to kind of take advantage of it. So to answer this question, how does density influence evolutionary rescue? It increases extinction risk and really prevents population growth. And then that high population growth is only happening under density independence. And it doesn't prevent adaptation. So adaptation is still happening. You just can't kind of see it in the phenotype of the population size. So the population density, that ecological thing, is may be shaping evolution because it might be leading to adaptation to those high densities. But the clearest signal we're seeing is that evolution in that eco-evolutionary circle is strongly shaping the ecology, the population dynamics there. In terms of conservation, I think what this means is that we have to think about restoring resources and habitat as well as keeping genetic diversity high if we want to have populations persist long term. Okay. And that is, I always talk faster when I'm up here, so we're doing good. That is the end of part 1. And now, so Gina asked me to talk a little bit about my role on this NSF Advanced Grant. So I'll tell you about what that program is and give you a little bit more background as well. And I love the science and the data, and hopefully this can be, it's important stuff too, and it can be as interesting as the science and the data. So first, I want to ask and answer this question, like, who am I to be talking about issues of diversity and inclusion in academia as a cisgendered white woman? So I'm a mom of 2 kids, 2 girls. I'm a mentor of lots of students. I'm a colleague. And around the time I started to think about some of these things more seriously in my workplace and what I could do about them was also the start of the Black Lives Matter movement. And this is one of my kids loves to draw. This is one of her cartoon characters. And I said, give me a gal that's mad, fuming mad. And she gave me this. And that made me mad and made me feel like I should do something about this. So part of what, as an academic, what I like to do when I want to like make some change is learn, right? So I went to, anybody been to NCORE? It's the National Conference on Race and Ethnicity in Higher Education. It is a fantastic learning experience, mind-blowingly different from a scientific conversation, conference, and I learned so much. The other thing I do when I want to learn about something is I teach. You know, like the best, like if you teach it, then you can know it pretty well. And so I taught some grad seminars first on women in science and then on gender and ethnicity in science to really understand what my social science colleagues were doing in their research, to understand that academic perspective as an academic to the core. I also took advantage of what CSU calls the Faculty Institute for Inclusive Excellence, taught by these 3 fabulous folks, Shannon, Rye, and Albert, and that was just super informative. And as like any good class or a learning opportunity like a master's or a PhD program, there was a final project that was required at the end. And I decided to do for my final project a seminar. Again, academic to the core. The reason I did this is because so much of what I had learned, other than through the graduate seminar, was about social and emotional effects and consequences of racism and sexism and other kinds of things going on in the workplace. And I just knew that my colleagues, as scientists like me, could like look at that and say, oh gosh, yeah, that's unfortunate. But to like really understand it, they needed to see the data. And so I thought, I'm gonna develop a seminar that is aimed at people like me who want to see the data, want to see some experiments or studies that really bring some of these things to life. And so I developed this seminar and I showed people things like for children taking tests, standardized tests to be put into gifted and talented programs. The test score is not the only thing that matters, of course. I mean, you can also ask about standardized tests, but that's another conversation. It also matters who the student is and who the teacher is. So Black children with really high scores can be less likely to be put into a GT, gifted and talented program, than white children with lower scores. And things like this were fascinating and devastating to me, because I had 2 girls in GT programs where they would have sessions for the parents on how special your child is, and they need lots of activities and engagement, otherwise they're going to act out in all these ways, and so we're going to create all these programs for them. Like, ooh, yeah, that's great for my kids, but what about for other kids who aren't getting into these programs, especially given things like this? When you look at severity of disciplinary actions here, comparing white and black children, these are, says students, these are younger than middle school. The first infraction, they're being treated approximately the same. And the second infraction, black children are being given much more severe consequences. And so poor little kids are bored out of their minds and acting up, and my children are being told, oh, you need more activities. And black children are potentially being told, no, you need to go to the principal's office. And so I tried to bring from birth on through getting to full professor data to my colleagues. And they found it really convincing, which was great. And that got me invited to a bunch of departments around campus to give that kind of talk. And I became the chair of a committee focused on the status of women on the faculty on campus. First on that committee, became the chair of that committee. We switched it to cogen from focusing on women so that we're really more explicitly thinking about gender equity on the faculty. When we created a program for mentoring women on the faculty, which I meant to say earlier, one thing that got me involved in equity work just from my own perspective was wow, I had no mentoring whatsoever. I didn't think to ask for mentoring, and my colleagues, that was very old school. My colleagues didn't think of giving it, and I'm okay, we need to change this. So we created this mentoring program in support of women in particular. So that led me to being asked if I could be PI on a grant to the NSF Advance program. So that program supported gender equity on, it's NSF, so really focusing on STEM disciplines for almost 25 years. It's been terminated. And my institution, Colorado State, had applied a bunch of times and not been successful. And finally, with me as PI, we received a 3-year award. And we were on a no-cost extension when it was terminated last spring. But I think we were able to do a lot of good before it was terminated. And I mentioned with me as PI because I really think that matters. Oh, but I'll get to that in a sec. I just want to first say, why does it matter to focus on faculty? Faculty members are we have a lot going for us, right? We're paid better than most at the university other than the upper administrators. We often have our own offices. We have a lot of power over what we do and when and how, power over students. And so it's this really privileged position. And also we experience all the shit that everybody else does, right? And there's a whole bunch of social science showing that when faculty members are successful and diverse, student success goes up and you get great scholarships. So if you want to be a fantastic R1 university, you can't just think about student success and research products. You have to really think about that. Something that links those together is, are the faculty members thriving? So I mentioned me not because I think I'm particularly that great at writing grant proposals, but I think ecologists and evolutionary biologists we're really good at working on our one little thing, mycorrhizae on something, and explaining to everybody why this matters to the whole world, to all of ecology and evolution, and then to kind of broader environmental systems. And so I think it really, it made it possible for me from outside the discipline to write a conceptually compelling grant. And then I also love data. And so I spent a long time working with folks at institutional research and inclusive excellence, getting data for this grant. Another thing is that we finally had really strong support from the provost and president's offices. So we were able to overcome this 20-year failure of getting the grant and finally getting it. And what we got is an adaptation grant. So you all had at University of Michigan one of the very first advanced grants, an institutional transformation grant that gives your advanced office here, was started by this program. We can't call ours advanced because it's advancement, which is development, which is fundraising. So advance is taken. But we have this adaptation grant. Nowadays, or before the program was ended, most of the grants given were adaptation grants. These are customizing evidence-based practices that all the institutional transformation grants, where you had social scientists studying how universities work and figuring out good ways to do things, they've already done a bunch of that. And now we can take those practices and customize them to the different universities that get the grants. So this is the research team, our grant writing team, and I just want to particularly mention that I had built relationships with Shannon when I took the Faculty Institute for Inclusive Excellence program, with Sue on Cogen, and Heather Novak. This is a classic. She and I go to the gym together. So I knew these folks and could call on them and push them in ways that is harder to do if you don't have a good, strong relationship with somebody. And with the support from the provost and president's office, we were able to get 3 new hires to the university who were first supported by the NSF grant and then switched on to base funds. And they also provided Mira Fa, who's a fantastic social scientist. Okay, so I'm going to present just a tiny bit about what we've done. And I really am flying through things. Talking about highlights of preliminary data, some of our key accomplishments, some challenges we faced, and then I'm director of our graduate ecology program, and so how I've kind of applied this within the ecology space. So highlights of preliminary data. This is, I think, a big reason that we got the grant is that I was able to get Heather to look at data on faculty in a different way. So they had previously looked at basically how the faculty population changed over time, but that was like people coming in, people going out, and you have this total population size. And I just said population size is really important in my evolutionary ecology part, but also that all sorts of things are going on within that population size that just the population you can't get at. So I convinced her to take this kind of cohort, more demographic perspective, where we gathered together essentially data on faculty members that had started over a broad period of time and tracked whether they stayed or left CSU for a 7-year period. And we could break that down with the numbers you get by grouping people together into 4 groups. So men and women, recognizing that gender isn't a binary, and then minoritized and non-minoritized people. And there we're following, this is racially and ethnically minoritized within the United States, following what NSF definitions. And that means that non-minoritized means white and international. So it's like these categories are not perfect by any means. They're masking all sorts of important different identities, and yet they're more than most people do in this kind of work where they're looking at gender and race as 2 kind of separate things, and you can't really explicitly look at that intersection between gender and race. So what the data showed when you break it out by all the disciplines is this. So these are also NSF kind of disciplinary groupings, which allowed us to get enough individuals in these groups to be able to track their whether they stay or leave CSU. And these numbers on the sides are how many people are in those groups. So you can see they're really small, and that in yellow, minoritized women are leaving they're leaving much sooner than other folks on the faculty. And you really can't see that if you group the groups together. And the patterns differ, you know, like in engineering relative to these two other STEM disciplines. But like the basic kind of shocker is the same. And this was really convincing to deans and to the president and I guess to NSF that like this is, you can't just say what happens in a department or in a college is, well, that particular person, they left because they wanted to, you know, be closer to their family back east. Or this particular person, they left because there was this or that other thing going on. When you see a pattern like this, you realize, well, like this isn't just an individual set of individual choices. This is a problem that CSU has that is leading people to choose to leave. And it's certain people over others. So we could also look at hiring. So I did this. It's really hard to tackle hiring data because it's hard to know what the pool is. And you advertise and you have your pool of people, and then the committee says, well, we're hiring, we're giving an offer to the best person for this, given the pool we have. And what we used for a pool here is earned doctorates. So NSF has a ton of data on who is getting PhD degrees or other kinds of doctorates in the US. And then we can show that if you look at the earned doctorates, so these are the folks who are in the pool, and then a period of search years after that, we're hiring more in the white men generally and international men than in these other groups, except for engineering, which had made some major efforts in diversifying their program. So again, it's pretty compelling that we need to think about hiring. So key accomplishments for the grant, I would say the data, like just having these data available is one of them. And so we have now a website where anybody can go to these, anybody who's an employee of CSU can go to this website, and it's pretty cool, I think. You can search you can look at ethnicity in more detail, or you can look, for example, so this is all departments, all colleges, everybody. You can look at retention through time of racially minoritized, non-racially minoritized, of gender alone, and say, wow, there's not a whole lot of big pattern there. But again, you look at that intersection and the same pattern falls out. You know, so the women of color are leaving more. So that is, I think, really good to have kind of there available for upper administration to see. And then we completely revamped our search chair training. And we modeled a lot of this on the way you all do it here, which is great. And one of the things we did is very, very simple. Just what used to be that the required search chair training lasted forever, and DEI in the search was optional. And now we have required search. The search chair training only lasts first 10, and then it will be 5 years. Laws change through time, and all of the material it's included into the search chair training. We created what's called the Chair Institute for Inclusive Excellence. So chairs and heads, like they have so much expected of them from all sides, and it's really challenging for them to do all things for all people. And so we try to provide them with some support, but we were pretty demanding, have been pretty demanding with, so these are 2 afternoons of 4 hours each. So it's a big time commitment, but they really, they say that I thought it was tremendously helpful, and especially they really liked being able to troubleshoot with each other, like, well, how do you do onboarding? And we give them a lot of suggestions for how to do these things and some evidence-based practices, but then they can troubleshoot with each other as well. And then finally, we created this Office of Faculty Success, which is like very similar to your advance. And we worked with faculty council on how non-tenure track faculty are treated and how so that they have clear promotion pathways and are called faculty. And we expanded that in-circle mentoring program. And there are other things as well, but these are just kind of like a few of the highlights of things we did. And then, of course, it doesn't all go well, right? This is hard work to do. It's not, at least for me, as joyful as collecting data on beetles, which I really love, but it's important. And then, so people get burnt out. People like me get burnt out doing it. All of us get burnt out on, oh my gosh, one more training. We don't have time for that. We want to do our science. So that's a challenge. There's also this phenomenon of, I don't know if you've heard this phrase, the left eats its own. I mean, like, since communism, the left has been we destroy each other. We have a very hard time, and again, like, there's a ton of social science that demonstrates this, like saying, well, here's our common goal. Let's work towards this common goal. We're like, well, but you said this and you said that. And we break each other down more than working towards our common goal. And that can be really, it's a real phenomenon, and it's hard, and you need to do some healing around that. The changes since January have been really hard, so we've had legal challenges. So we used to have this Encircle program was in support of women, and then I forgot to mention when we adding it to part of faculty success is we expanded it to have in support of women and gender non-binary folks of color. Fort Collins is super white, and so it has been really meaningful for faculty members to get together and have just folks they can talk about their experiences in Fort Collins and at that university. And literally several of them have said, I would have left this university if it weren't for this program. But now, and we use the language of in support of to, whoops, it did go, then I pressed it again. To get around needing to include everybody. So anybody can go to it, like a white woman could go to this mentoring circle in support of women of color or folks of color, but now we're no longer able to do that. And we're having to spend a ton of time working with CSU Legal to make sure that things on the website are in compliance with what they see as new interpretations of laws. And so that is all kind of exhausting, and now the Encircle program is very general, and we'll see how it goes this year. It might be effective, might not. Our state is also, I know that we all feel like universities are going broke everywhere. We're vying for 47th or 48th in the nation every year for support of higher education because of the way the Colorado tax structure works. And so money really does become very limited, and the big, beautiful bill has reduced that funding even further. So those are some of the hard things about doing this work, and especially in this time. And there's also costs and benefits for scientists like me doing this work, time away from the science, really reduced productivity for sure, some of that emotional strain. But it's so rewarding, and I've been able to use a bunch of what I've learned as director of the ecology program. I came in and there was kind of nothing going on for inclusion and mentoring and diversity, and it was just super easy. These are it's not like picking the low-hanging fruit. It's the apple's right there on the ground for you to pick up, and so you can just make some changes really quick, and that's been super satisfying. And with that, yeah, thanks. And here's some reminder of the data I showed. Any questions on science or other? You probably don't see talks like that often. Yes? Oh yeah, do you want, should I hear I can throw this? I'll toss it partway and then.
So in the your second part, you talked about how, as evolutionary ecologists, we like to collect data and make our proposals like that. So with, you know, what you're doing at Colorado State, what data are you interested in collecting? Like, I'm hoping this is something that you can collect for a long time to really kind of show that these efforts that we're doing are making impact across scale with students, with faculty. I guess if there's anything that you want to share based on like that.
Yeah. I mean, so I think that some of the changes, like IR having the faculty retention data there publicly and so easy to look at different kind of slices of the university and different identities, that we can tell from that through time, does that change? And then they, of course, also collect data on student retention. It's really difficult to link that directly to our efforts on faculty. And then data that I didn't show you are on our climate survey. And so those data are collected again. And then as a scientist, it's hard for me. I'm like, well, so much has changed. Now we have Trump in office. Now we have this, like, all sorts of other things going on. Climate has changed in other ways. How much of it is what we've done and versus all the rest of the changes in the world? But we will have that information. There will be questions focused on some of the kinds of things that we're doing. Yeah, but it's really hard to link. And I think the social scientists working on the project who have interviewed and collected statements from folks who have been part of particular programs that have been put on, I mean, people are finding that really meaningful, and the social scientists, you know, those are the data for them, and so it's a short-term change, but they're finding those programs really meaningful. But yeah, it's hard work to do, and it's hard to know, like, well, did it do anything? Did it work? Good question. Yeah.
Ask it without the mic.
Yeah.
How about I ask it, you repeat it?
Okay.
So I was really interested in the evolving population
Uh-huh.
of beetles that you have there, and the density and dependence. That population just increased dramatically in size. And I immediately thought about invasive species and invasive plants. What sort of, like, I wonder if you could relate that to the well-known 50-year lag in where you have like an introduced species and it takes 50 years for it to sort of.
Yeah. So Gina is asking about these data, especially with density independence and in invasion biology. A time lag is often seen before when something's introduced. It's hanging down at low levels, and then all of a sudden it takes off. And I think that, so I have done experiments with Tribolium where we put them in arrays. So instead of like groups of boxes, we put them in arrays. And so they're invading out through this system of arrays. And when they can evolve, they go faster, they go further, and the population size gets bigger. But there's also that, you know, it does take them a little while to adapt. And so that adaptation process is, I mean, I think it matches with a lot of the data on species are around for a while. And exponential growth takes a while. At the very small sizes, population sizes, even if it's just ecological, you can't see them for a while, and then it shoots up. And then two, you add into that it takes time to adapt, then yeah, for sure. That could really help explain those lag times. Either one.
Okay. I have a separate question about a different term of the same graph. have the genetic toolkit to actually make it faster. And it's interesting that it's actually fairly well crammed together. They're very consistent.
Yeah, it is. So I'll show you the really messy, where is it? Here. No, I have here some very messy but real data of individual populations. So hopefully this gets, I should have repeated the question. What are sample sizes, and are there at least some populations in that bottleneck density independent group that can grow more? And so, yeah, the answer is yes. There are two of those that are shooting up exactly. And that, yeah, the modeled results, you can't see that. And I just decided I didn't have enough time to go through. I'm like, oh, I better hide some of these. But yeah, really good question.
Very cool. Thank you.
Yeah.
We talk about the consequences, thinking about the invasive species. But I'm curious if your results can also be applied for the conservation, like repopulate communities and.
Yeah, I think very much so. It's really direct. So the question is, so I've talked a little bit about how it relates to invasive species, but what about for conservation? And I think that it really does mean, I mean, so you've probably heard of like translocations or kind of genetic rescue. I think that we do need to be doing that. We need to make sure that populations of conservation concern are, like, we're maintaining their genetic diversity somehow so that they can keep adapting to changing environments. And then we can't just say, okay, well, that's good. We have to be figuring out ways to get them more habitat and space, because when the population stays small, there's just still going to be at risk of extinction over the long term. So yeah, I think it's super relevant. Anything else? Yeah, Gina?
So this new provost-funded faculty success program, talk about that a little bit. Are you going to lead it? Or?
Hell no, I won't go. Sorry if I'm being offensive. That was supposed to be funny, but y'all are a hard audience. It's great, and I am continuing to help it, and we're trying to hire an associate vice provost to help the vice provost with the whole program. And I am not interested in applying because I'm kind of burnt out on that work, and I can help them however they want, and I want to do more science. It's good stuff to do, and I really highly recommend getting involved. And then you have to know your limits, and also it's not necessarily my strength, you know. So there are some other really great people who would be good at that job. Yeah.
Just a quick methods question with the density independence. So density is influenced by space and resourceability, right? So when you were increasing the size of the box as the population size increased for the density independent treatments, was that also increasing the amount of food available to them?
Yeah. So here, it's simplified in the Tribolium system because the food is the habitat. It's actually, even though the cartoon showed the box growing, but it's actually, we're just adding more boxes, and then they've got holes, precision holes drilled through them so that you can have movement among boxes. But yeah, so the amount of food is increasing with the amount of space. Yeah, good question. All right, I think you've earned your Thursday afternoon. Thank you all so much, and thank you, Gina, for inviting me. Yeah.
What you do with tarantulas.
Oh yeah, I should shout into this thing.
see that, and so in South Africa there's another genus, so these are like the free known genera of inhibits. So all